A very big hello to all my wonderful PU2 and KCET champions. I hope you are doing absolutely great. So in today's video, we are going to deal with a chapter that might have scared most of us in this 12th grade, and that is Principles of Inheritance and Variation. Now some of you might be thinking, ma'am, it's not that difficult. Okay, it not be that difficult for you, but for most of us, it had been. For example, when I was in 12th grade, I remember being scared of from this chapter. But now I'm not in 12th grade anymore, so you can be safe because I am going to teach this chapter very nicely to you so that you understand each and every concept. And while watching this, if at all you feel that, okay, this video was good, so you should be definitely clicking on the subscribe button as well. This chapter gives you 13 marks in your PU2 and almost 4 to 5 questions in your KCET examination, which means that we have to be very careful. And if at any point you get bored, or basically your concentration goes somewhere, pause the video at that point itself, listen to what do you call a song or maybe stroll around, and please come back, okay? Come back and watch the entire video. Wishing you all the best. Let's go ahead with the first thing that we have, and that is Mr. Gregor Mendel. So Gregor Mendel, my dear children, is the pioneer of genetics. We call him the father of genetics. Why? Because he was the one who discovered the basic laws which are there, and somehow it has been taken into consideration now also. And his experiments were exclusively on a plant which you see right over here. It's known as pea plant. And you also call this as garden pea. You also call this as what? The scientific name of this is Pisum sativa. Pisum sativa. So please have this in your memory. And you know what? This person was so much dedicated that he spent 7 years experimenting. Just imagine 7 years of your life. By this 7 years from first standard, you reach to 7th standard, right? So it's very long period. And what was the period? You need to remember this date. That is from 1856 to what? 1863. Those 7 years, my dear children, he worked on this particular plant, that is garden pea. So if at all you remember 1856 and then just add 7, it will be 1863. So you do not have to remember 2 of the dates. At least this would be more than enough. Got it? Fine. Now, why did people take his works into consideration? Because he was a person who started using mathematics in biology. So now when I come and say that this much percentage of things happen because of these things are there, whenever percentage or whenever something analytic related thing comes up, we all start believing things, right? So similarly, he was the first person to use mathematics in biology. He gave data, statistical analysis, which made it more, trustable, right? Second thing, he had a large sample size. Large sample size as in he didn't work on just a few things out there. No. For making sure that his experiment was correct, he had a large number of plants up there. He breeded them repeatedly, and that's how he got his conclusion. And as I said, this point is linked with the other point, that is repeated experiments. So yes, he had large sample size, and apart from that, he repeated his experiments so many times just to make sure that everything that I'm going to tell is right or right itself, right? So that, yeah, so basically that was being done. And now the fourth reason is that he chose a plant that had clear and contrasting traits. What do you mean by that? So basically, he chose the plant that is what? Pisum sativa, I told you. And when I'm saying that it has clear and contrasting traits, I mean that basically in this particular plant, there were 7 characters. Okay, characters as in, for example, flower color, plant height, seed color, seed shape, pod color, pod shape, and flower position. So there are multiple characters. In these characters which he studied, there were certain traits also. So character, for example, when I say height, I have tall and dwarf. Height is a character. Tall and dwarf is what? The types of this particular character. I call them as what? Traits. What do I call them? Traits. So over here for flower color, he had purple and white. Aren't these contrasting traits? Yes, my dear children. And basically with the help of these traits, he get better conclusion. He used to get what? Better conclusions as well. So over here, ma'am, do we need to remember these 7 contrasting characters out there? The answer is yes. Do we need to remember the traits also? The answer is yes. How can you remember? The way is simple. Okay, so all of them are having a pair. For example, if at all you talk about flower color, okay, if at all you talk about flower color is there and flower position is there. Flower color is there, flower position is there. In flower color, you have, as I said, purple colored flower and white colored flower. Which one is going to be the dominant one? Which one is going to express? Suppose both of these characters, traits, genes we are having. Which one do you think will be expressing over the other one? It will be purple color. So I'll call this as dominant traits. Do not worry, I'll talk about it in a bit more detail as well. So purple is dominant, whereas white, which you see, is what? A recessive color. Now over here, purple can be also told as violet. Do not be confused. Okay, so flower color, you had violet as well as white. Flower position, you had two things again, that is axial and terminal. Which one? Axial means what? Axial means, suppose I have a place from where I will get a new branch, but here I have a flower. This positioning of flower is known as axial positioning. Suppose I have a branch, at the tip of it I have a flower. This is known as terminal position. Which among this was a dominant character in the case? In this particular case, it was axial. So you can remember A comes before T, right? A is the first alphabet. So A is the dominant one. So that is about flower color as well as flower position. Now you have seed color. Okay, let's go to seed color. Seed color and seed shape. Okay, seed color and seed shape. Now, usually when you check up the pea plants, we eat the green pea out there, right? But you know what? Yellow color is actually the dominant one. And green color, which is there, it is the recessive trait out there. Okay, now what about seed shape? Seed shape can be like this, round, or sometimes it can be like this, that is wrinkled. So which one is dominant? We know it is definitely round, which is going to be dominant, and wrinkled is going to be recessive. Okay, so flower color, flower position done, seed shape and seed color done. Now what do we have? We have pod. So basically the green peas which are there, they are covered inside a structure like this, right? So this is known as pod. In pod also you have pod color and pod shape. So pod color, so usually when you go to the market, you see green ones everywhere, right? So you can remember the green one is dominant over here. So previously in seed color, yellow was dominant, but in pod, my dear children, green one is definitely dominant and yellow one is going to be the recessive one. What about the pod shape? Pod shape, as in it can be full like this or it can be like this. You call this as constricted. What do you call this? Constricted. So basically inflated or full is the dominant character and constricted one is going to be what? The recessive character out there. Got it? Fine. Now the last one that we have does not have any pair, and that is plant height. And plant height is the easiest one to remember. It has tall and short plants. Tall one is going to be the dominant trait out there. Got it? Fine, ma'am. Yes. So how do you remember this? You can remember it like this, and I'll give you a code also. Gravity. This is for all the dominant characters out there. Okay. Green as in pod color. Okay. Round as in the seed shape. Axial as in the flower position. Violet as in the flower color. I is for inflated, that is the pod shape, and T which is there, T is for what? Height, that is tall, and Y is for yellow, that is a dominant trait in the case of seed color. So these are the things that you can remember. All the dominant characters are there, but do not get confused with green is dominant in seed color or pod color. So please understand, green is dominant always in pod color. Okay, you choose the green parts. Fine. So this is done. You can get questions on the basis of it. You had got questions in KCET. You can get questions in your PU2 as well. Okay, they can ask you which one is not the character that was studied by Mendel. Now coming up to what he said. So basically, he did an experiment when he crossed tall plants with dwarf plants. The tall plants, my dear children, when I say it's tall, basically you need to understand that over here, there are 2 chromosomes for each of the character that you see. 2 chromosomes. Okay. One we get from one parent, another we get from another parent. And on this chromosome, at the same position, my dear children, you would be having the gene for that particular character. Now over here I see it is capital T gene, and here also it is capital T gene. That means both these genes are same. You call this condition as what? Homozygous condition. Homozygous condition. Why homo? Because it's both of them are same. Here also. So homozygous, what is it? It is tall. It is what? Tall. Here also for tall we see, we have at the same position the genes for height. Okay, but these genes are different. These genes are what? Small t. Here also both of them are same genes, only small t. So I'll call them homozygous. Homozygous what? Homozygous dwarf. They are short, right? So I'll write homozygous dwarf. So what did you basically understand from this? That basically for height, if at all you see, you have two genes. One is capital T, other one is small t. These two type of genes which are there, I will call them as alleles of a gene. The types of a gene, I'll call them as what? Alleles of a gene. So the gene for height has two alleles. One is capital T and other one is what? Small t. Got it? Fine. Now, listen to me very, very carefully. So over here, I had the two chromosomes. Here also I had the two chromosomes. These are homozygous tall plants. These are homozygous dwarf plants. I can also call this as pure breed. I can also call this as pure breed condition. That means when they have both the alleles as same thing, you call it that. Now, my dear children, from this, only one can go into a gamete. Why? Because gametes have only half the number of chromosomes. Because suppose egg is having 23, sperm is also having 23, 23 gives us 46. 46 was a normal number of chromosome, but only half came, only 23 came. Why? Because anyway this egg and the sperm are going to unite and give rise to the full number of chromosomes out there. So anyway we have to make sure from each chromosome pair, you have multiple chromosome pair, from each chromosome pair only one can go. So here whatever you do, you will anyway get the capital T allele. Right? Because both the chromosomes have capital T only. Similarly, from here also one will be coming up, that is this. Now, a egg is carrying this, a male gamete is carrying this. When both of them are fusing, again you get two chromosomes. But over here, if at all you see, one is capital T, one is capital T and other one is what? Small t. Right? In this condition, which character is going to get appeared? Which trait is going to be appearing? So here it was tall, here it was dwarf. When both capital T and small t alleles are coming, which character is going to appear? The tall one is going to appear. Why? Because the tall gene which is there, or the allele which is there, it is dominant. It is able to produce more proteins, which is why the character that is going to be seen is what? Tallness. What do you call this condition where both the alleles are not same? Until now we studied homozygous condition where both the alleles were same. This is not homozygous condition. This condition is known as heterozygous. Why heterozygous? Because both the alleles are not going to be the same, my dear children. I hope that works. Yeah. So in the first generation, so you had the parents, you had the gametes, and this is the first generation which you also call as what? F1 generation. You got the dominant trait. You got the dominant trait which was what? Which was tallness. Okay, fine. So dominant trait was only visible. So Mendel, when he was checking the plants, he was like, all of them are tall. None of them are dwarf. That means the genes didn't pass. He was confused like that, which is why he went again and he did his actual experiment. So basically he got multiple plants like this, capital T, small t, capital T, small t. He crossed them. Okay, he crossed these plants. This is known as selfing. It is known as selfing. And what did he get when he did the selfing process? Let's see that. Just give me a second, okay? So, where are we? Over here. Okay, so when he did the selfing, it was capital T, small t parent, capital T, small t parent, right? As I told you, this is what the parent. Now I want to form the gametes. The gametes can be either this, half of them is going to go anyway. So some of the gametes can carry capital T, some of the gametes will be carrying small t. Similarly here also, some of the gametes will carry capital T, some of them are going to carry what? Small t. This is how it goes. Now after gametes, you want F2 generation. This is what? The filial 2 generation. So just a second, there is some glitch out there. Okay, these were the gametes. Now in F2 generation, what is the thing that you're going to get? Suppose this gamete and this gamete came together. You got capital T, capital T. When this and this came together, you got capital T, small t. When this and this came together, again you got capital T, small t. Always write the capital letter first. And then when you cross this and this, you got small t, small t. Now ma'am, out of them, I can say this is definitely going to be tall because this is homozygous condition. This is also going to be tall because we consider this as tall, right? So this is going to be dominant over the small t, which is why these are also tall. So 3 of them are going to be tall. And Mendel was surprised because he saw a dwarf plant in the next generation. He saw a dwarf plant in the next generation. So out of 4, 1 of them was dwarf and 3 of them were tall. So he got to understand that yes, it didn't appear in the first generation. Probably there was something that was dominating over it, which is why it was not appearing. But the gene was anyway there, or the allele was definitely there. That is why it was appearing in the F2 generation, filial 2 generation. Now, when you say that this is tall and this is dwarf, you call this thing as the phenotype of the plant. That is the physical appearance. Okay, phenotype means physical appearance. And if at all you're asked this, that please give me the phenotypic ratio. How will you give the phenotypic ratio of this particular thing? So 3 of them are tall, and 1 of them, my dear children, are going to be dwarf. So your ratio is going to be 3:1. And whenever you write 3:1, make sure what is 3 and what is 1. So 3 of them tall, 1 is dwarf. Okay, there is one more thing. This is about the physical appearance, right? Now, what if we talk about the genotype? Ma'am, what is a genotype? Genotype means the genetic makeup. So 1 is capital T, right? 1 is going to be, just a second. 1 is going to be capital T. Okay. 2 of them, Baba, are capital T, small t. 2 of them, capital T, small t. And 1 of them, which is that, is what? Small t, small t. So that is what the genetic ratio or the genotypic ratio, which you call as 1:2:1. So if at all you are asked about what was the ratio, phenotypic ratio in F2 generation in a monohybrid cross? Why do you call it monohybrid? Mono means a single character is something that we are studying. Hybrid means basically from capital T, small t, when you combine, you got a hybrid that is having 2 different alleles. So you call this cross as what? Monohybrid cross. So the ratio of monohybrid cross is what? Phenotypic ratio is 3:1 and genotypic ratio is going to be 1:2:1. This is super important piece of information. Okay, and here itself I'm going to talk about 2 laws. Listen to me carefully. The first law is law of dominance. Okay, so after this experiment on Alfred Sturtevant, the monohybrid cross experiment, Gregor Mendel came up with 2 laws. One is law of dominance, according to which he said that if at all there are 2 pairs of factors, okay, or if at all there is a pair of 2 different factors as in alleles. At that point he did not know that what are genes and things like that, so you used to call them as factors. So basically when you have 2 pair of dissimilar factors out there, they are not similar, they are dissimilar. One is going to dominate over the other one, and the one which is dominating is known as the dominant allele or the dominant factor, and the one which is not able to express itself, which is small t, will be known as what? The recessive factor. That is known as the law of dominance. So in a pair of dissimilar factors, one is going to dominate over the other, and that is going to be expressed, right? Now there is one more law, that is law of segregation. What does law of segregation say? Law of segregation says that when gametes are being formed, when I see the gametes are being formed, from a pair of factors, only one factor goes into the gamete. They are segregating, right? They are segregating, and from a pair of factors, only one factor goes into one gamete. Another factor will be going to what? Another gamete out there. It's not like 2 factors can come into the same gamete. No, they are going to segregate. This is known as law of segregation. I hope that's clear. If at all law of segregation was not there, you will not be able to see this ratio. You get it? So law of segregation means in a pair of factors, all factors are going to separate and move into gametes. Now there is a condition where where there is homozygous condition, that means the pair factors are similar. For example, capital T. In that case, the gametes will also be having the same type of alleles, that is capital T, right? In a pair of dissimilar factors, you get different type of gametes, one capital T and one small t. Got it? That's all. Okay, that's all. So this is an important ratio. Please remember it. This is a ratio of monohybrid cross. Now, everyone who is preparing for KCET examination, I hope you all know that we have launched a ABHYAS KCET test series, and by if any chance, if at all you have not registered for that, go register right away, because this is super important test series for you. We are giving you 35 KCET mock test. We are giving you chapter-wise KCET PYQs also. So this is a new edition that we have, and all of it will be under what? A timed condition. So you will have a timer. You have to give your answers according to timer. This is to build your speed, okay? And make sure you take it up, because my dear children, so many of them took it the previous year and they got the best benefits of this. Okay, we got rank holders with the rank 171, 443, 693 and so many. So make sure you check it out once. It's not like necessarily you have to take it up. No, check it out. The link will be given in the description section. You can ask to your seniors also. And the pricing for this is rupees 149. So if at all you're planning to buy, take it up. Do not go ahead with mock test now because you do not have time for that. You can do the mock test later on. At least by now you can go ahead with chapter wise KCET questions. So once you're done with this chapter, if at all you're preparing for KCET, go ahead and solve the pre-YQs. If at all you're just preparing for your PU2, we have already recorded free videos, Brahmastra videos. Okay, go watch it. It will be really helpful for you. Cool? Yes, ma'am. Fine. So now going ahead, all about the Mendel's law of inheritance. So basically we spoke about all of that. Okay, want to know the findings of his experiments. Let's see what did he find after the experiments. So Mendel proposed that something is stably passed down from parent to the offsprings through the gametes over generation. Something is getting passed from the parents through the gametes to what? The offsprings, to the babies out there. What is that something? Genes. But did he know it was known as genes? No. What did he call it? He called these things as factors. We call this as what? Genes. Okay. Genes are the units of inheritance which are there. Yes, genes are unit of inheritance. Yes. Genes carry information required to express specific traits in an organism. When I say a gene, my dear children, I talk about the segment of DNA. This segment of DNA is going to code for some protein, and this protein is going to make something else which is going to define our character. That is why we say that genes are units of inheritance. They define all the characters that we have. Now, alleles are different forms of the same gene that code for a pair of contrasting traits. By that I mean, for example, when I say there is a gene for height, there can be capital T, there can be small t. Both of them are genes itself, but they are two different types of genes, which is why I'll call them alleles of a gene. Types of a gene, or what we call as alleles of a gene. He proposed that in a pair of dissimilar factors, one dominates the other. So in a pair of dissimilar factors, what do you call this condition as? Heterozygous condition. One is going to dominate over the other. The one that dominates is known as dominant factor, and the one that is not able to express itself is known as what? Recessive factor. He also said during meiosis, allele pairs segregate and only one is passed to each gamete. So as I said, when meiosis is going to happen, the alleles pairs which are there, they are going to separate and only one is going to be passed into one gamete. Done? Done. So he gave law of dominance. Characters are controlled by discrete unit known as factors, and factors occurs in pairs, you know that, yeah? And in a dissimilar pair of factors, one member will be dominating. We studied this. The law of dominance is used to explain the expression of only one parental character. So this is a very important MCQ question. They ask you, the dominant allele expresses in F1 generation and F2 generation. Is that true? Yes, it does. We saw tall plants in F1 and F2 also. Okay, what about the recessive trait? Recessive traits are only going to appear in F2 generation. Recessive traits are only going to express in F2 generation if at all you started off with these type of parents. If they were homozygous tall and homozygous dwarf parents that you took, or pure breed tall and pure breed dwarf you took in the first generation, then yes, in the second generation only you will be seeing the recessive character. You saw that right now, 3:1 ratio. One was dwarf, right? So this is a very important point. If at all you are crossing pure breed tall and pure breed dwarf, in the first generation you will only see the dominant parent. In the second generation also you will see the dominant parent, but the recessive parent are going to express only in one thing, that is F2 generation. Okay, this is the case when these are the parents. If parents are different, the things will be different. Please make sure you check it up and don't only go for answering the question. Okay, this was the first law. Second law is law of segregation. Alleles do not blend. Both reappear unchanged in F2 generation. Yes. Alleles, do you think they are blending out there? Tall and dwarf, they are blending to give you a medium character? No. So alleles are not blending. They reappear unchanged in the F2 generation. So the first generation when dwarf character was not there, Mendel was like, where did it go? Did it blend? No, it was not. Okay, now during gamete formation, allele pairs separate, so each gamete gets only one allele. We discussed about all of this thing, right? Hey, homozygous parent produce two gametes, identical gametes. Heterozygous parents produce what? Two type of gametes. Done. Easy, easy. Okay, now, is there a easy way to cross? We did this crossing and all, right? So he is saying, yes, my name is Reginald Punnett. I am a British geneticist, and I know that's too much of information. Yes, that's actually too much information. We just need to know his name is Reginald Punnett, my dear children, and yes, he was a British genetist. You need to know that because you get a question like that also. Okay, so please make sure you know he's a British geneticist, and his name is what? His name is what? Reginald Punnett. And what did he come up with? He came up with Punnett square. What is Punnett square? So basically when I'm crossing things like this, it's kind of difficult for me to do this, right? So he came up with Punnett square, where we just need to do two things. That is, put the gametes of one parent over here. So capital T and small t. Okay, some gametes will be having capital T, some gametes will be having small t. Similarly, some gametes will be having capital T, some gametes will be having small t. When you fuse this with this, you get capital T, capital T. When you fuse this with this, you get capital T, small t. When you fuse this with this, again capital T, small t. When you fuse with this, you get small t, small t. So three of them are tall. I can clearly see that. Two of them heterozygous tall, one homozygous tall, and one is going to be dwarf. Yes, this is much easier and a cleaner way to do this, and this was given by Reginald C. Punnett. He was a British geneticist. Okay, done. Fine. Ma'am, I have a doubt. Okay, how did Mendel get to know if the plant is tall like this or this? Fair question. If you see a plant, how can you tell that it is homozygous tall or is it heterozygous tall? Fair question, right? So my dear Papu asked me this question, which is why I need to answer it. The answer is what? Test cross. In this condition, you do a cross that is known as test cross. And for those of you who have no idea about test cross, listen to me very, very carefully. So it goes like this. Here, whenever you're confused whether the parent is showing the dominant character, okay, dominant character as in tallness, right? We do not know. They show, when the parent shows a dominant phenotype or the physical type out there, we can confirm what is their genotype with the help of crossing them with the recessive parent. Recessive parent means this is a recessive parent that we have. And how do we cross it? Let's see how do we cross it. So this is known as test cross. Okay, so suppose I am unsure whether it is capital T, capital T. And on this side, just a second. And on this side, I'll put capital T, small t. Okay, so I'm crossing it with what? The recessive parent, that is small t, small t. Here I have capital T, capital T. Fine. Similarly, I'm crossing this with what? The recessive parent. Let's see what is the thing that you are going to get differently. And with how can you identify whether the plant is like this or this? When you cross this with this, you get capital T, capital T, small t. Here also capital T, small t. Here also capital T, small t. Here also capital T, small t. What do you observe? You see everywhere you have one capital T. That means all of them are going to dominate over the other, and all the plants are going to be tall. Okay, fine. What about this? I do this capital T, small t, capital T, small t. When this and this is crossed, small t, small t. When this and this is crossed, you get small t, small t. Okay. Now, if at all I see, I get two of them which are dwarf and two of them which are tall. So when I get all the plants to be tall, I understand that this plant which was appearing tall to me is actually homozygous tall. And if at all I get dwarf and tall plants, I basically understand that this plant which is appearing tall to me, it is heterozygous tall. How did you get it? By crossing with a recessive parent. You have to cross with a recessive parent. This is known as what? A test cross. So in a typical test cross, an organism showing a dominant phenotype, that is tallness here, is crossed with the recessive parent instead of self-crossing. So instead of doing self-crossing, you do this. You get it? Because here my dear children, yeah, that's all. I don't want to complicate this. This is known as test cross. And in test cross, if at all you have noticed, you are crossing with the parent. That means you went back and you crossed with the parent. This is also an example of back cross. So test cross which is there is a type of back cross. In test cross, you are going back and crossing with a recessive parent. Back cross is basically just going back and crossing with any parent. When you are specifically saying that you are crossing with recessive parent, you call this as test cross. I'm telling you again, back cross means going back and crossing. Here isn't it happening? Yes. And here you are specifically crossing with what? A recessive parent, which is why you call this as test cross. Okay, fine. So yeah, this is an example given in NCERT. Violet flowers are seen, you're not knowing whether it's like this or it is like this. So what do? You cross with the recessive parent which is there. So violet is represented by WW, capital W, capital W. This is different from what we usually assume. We always take the dominant character's letter, that is V. But over here, W has been taken. So please consider that. It's not a big issue. Okay, capital letter mean dominant character. That's all. Okay, fine. Now, test cross versus back cross, did I explain you? Yes. Crossing an individual showing a dominant phenotype with its recessive parent alone is known as test cross. Crossing an F1 hybrid with either of its parent either of its parent. Okay, anyway, it's going back either of the parent, that is known as backcross. Done? Done, ma'am. Fine. Let's go about the exceptions of Mendel's law, and this is one of the favorite part of this particular chapter. So Mendel did so many experiments, but there were some exceptions also. Not every time whatever he said came out to be true. Okay, there were some exceptions. Exceptions are always there. Haven't you heard of that? Yes, right? Just give me a second. I'll just tie up my hair. It's getting hot here. By the way, are you enjoying the class, or is it like boring for you? I hope you're enjoying, and do not judge me on the base of my hairdo out there. Yes, let's go. So over here, the first exception that you have is of what? Is of Snapdragon antirrhinum. Yes, Snapdragon, or you also call it an antirrhinum majus. Do not be worried. This particular plant is super important for you to study. Why? Because this is going to break all the trends, right? So here I'll tell you the funda, the basic thing which is there. There is a plant that gives you red color flower, and there is a plant that gives you white color flower. Both of them, they are snapdragon flowers only. Okay, so now what do you expect when you cross this and this? What do you expect? Basically capital R from here and small r from here. When I cross this with this, I get capital R, small r. Oh, capital R is there, that is a dominant character. Obviously, this plant is to be red. But you know what? It is not red. Instead, my dear children, it turns out to be pink in color. And that's how it became an exception. Mendel told that pair of dissimilar factors, one is dominate and show the dominant phenotype. But here it is showing another phenotype that is pink color. People got shocked. So they thought, why not do selfing? Let's cross this plant with this plant itself, and let's see what happens in the next generation. So that was being done. Let's see what is the conclusion that we got. Capital R, small r, capital R, small r. Here you get two type of gametes. One is capital R, one is small r. Here also two type of gametes. One is capital R, one is small r. When you cross this with this, you get capital R, capital R. When you cross this with this, you get capital R, small r. When you cross this with this, capital R, small r. When you cross this with this, small r, small r. Three of them should be red. Why? Because you have the dominant allele over here, right? But you know what? Again, a shocker came into the picture. This was definitely red. These two were still pink. Were still what? Pink. What about this guy? This was white because there is no one to dominate, so it is smaller, smaller. Why do you think that happened? Because the alleles over here, they are not completely dominant, or the dominant allele is not completely dominant over the recessive allele. You call this condition as what? Incomplete dominance. Incomplete dominance, and this, my dear children, is an exception that you have. And if at all you are asked, what is the phenotypic ratio? Phenotypic, ma'am? Physical, right? PP, physical feature that is there. One red, two pink, and one white. One red, two pink, and one white. This is what the phenotypic ratio. Okay, in that the phenotypic ratio was 3 tall, 1 dwarf, that is 3:1. Here it is 1:2:1. Did I put 2:1? Just a second. Yeah. Now, going ahead with the next thing. What is the genotypic ratio? One is capital R, capital R. Two of them are capital R, small r. And one of them is small r, smaller. Again, you get what? You get the ratio which is what? 1:2:1. Why am I doing this thing? Just a second. Okay. So here the phenotypic as well as the genotypic ratio is the same, and that is 1:2:1. Got it? And this is incomplete dominance. You see it in what? Snapdragon or 4 o'clock plant, or also you call this as antirrhinum species. It is known as antirrhinum majus. This is your first exception which is there. Fine. This is done, and this is the, by the way, this is the snapdragon flower which you have. Cool. Now we discussed about this. Incomplete dominance is a type of inheritance, but neither allele is completely dominant. Neither of the alleles are going to be completely dominant over the other. The result, they often show a blending of traits out there, okay, which was not usually being seen. Now, the next exception that you have is multiple allelism. So according to Mendel, he said that there are pair of alleles, pair of factors. For one character, pair of factors. That means height, then tall and dwarf. That's all. But now, people started observing what? Multiple alleles for the same character. For example, in blood groups. In blood group, you have 3 alleles. One is IA, another is IB, and the third one is IO. Are they an example of multiple allelism? Yes. This is the second exception. So after incomplete dominance, multiple allelism, where there are multiple alleles governing a character, this is known as multiple allelism. You know there are 3 alleles for what? For blood group. Okay, this exception is also done. Now the other exception is related to this, and that is codominance. So I'll tell you the funda first. Listen to the funda. Suppose this is a cell. This is a red blood cell. The plasma membrane of the red blood cells, my dear children, has sugar polymers that are going to protrude from its surface. And the kind of sugars which are going to be there are determined by the genes. By the genes. And as I told you, you have multiple genes. IA, IB, and IO. These genes, whatever gene combination comes up, that will be deciding what sugar polymers are going to be there present on their plasma membrane. Fine, fine. Let's talk about what is codominance and what is going to happen in general. So over here, I need few things. Okay. When I say someone is having a blood group, I mean that they have 2 alleles and both these alleles are IA, IA. Or there can be a condition where one allele can be IA and the other allele can be IO. What do you understand from this? That A is dominant over O, which is why they are showing what? A blood group. Now what about B? Similarly, it can be IB, IB, or it can be IB, IO. Ma'am, does that mean that B is also dominant over O? The answer is yes. Since O is definitely a recessive allele that you see, it will only express itself when both the alleles are same, that is, it is IO only. Done. Because anyway someone else come, it will be dominating that. Ma'am, there is one more blood group that you are missing, and that is AB blood group. Okay. In AB blood group, the trend is seen, that is, it will be having one allele for IA and another allele for IB. Both the alleles are there, my dear children, and both of them are going to equally express, which is why I'll say it is what? An example of codominance. Codominance. Both the alleles are expressing themselves. This is what? Codominance. Please do not forget it. And always remember, IO is sometimes written as I, like this small letter I, not be confused. Okay, this is codominance. We have never seen that only one is going to be dominant, but over here both of them are equally dominant. Fine. So yes, the plasma membrane of the red blood, I told you, but when IA and IB present together, they both express their own phenotype, that is their sugars, which is why you call this as codominance. So over here, you have to know about when is IA coming, when is when is A blood group coming, when is B blood group coming, when is AB blood group coming, when is O blood group coming. Everything you need an idea of it. How many alleles are there? 3 of the alleles are there. How many maximum phenotypes are possible? A blood group, B blood group, AB blood group, O blood group. So how many phenotypes? Basically 4 of them are possible. How many genotypes are possible? For A we saw 2, for B we saw 2, and 2 plus 2 4, and then for O we saw 1, that is 5, and AB 6. Okay, in AB you have IA and IB. Right, so 6 genotypes are maximum possible and 4 phenotypes are maximum possible. Okay, 4 phenotypes. You might be given question that when these alleles are there, what is the maximum phenotypes you can observe? What is the maximum genotype that you can answer? So please tell that, do the calculation properly and then only go ahead. Okay, fine. So if at all I am saying that both the parents, okay, just to make you understand, if at all I am saying both the parents are having AB blood group, what is the maximum phenotype that you can see? Okay, both the parents are having AB blood group, right? So in that IA, IB, IA, IB. I need to cross it. When this and this comes together, I get IA, IA. That means A blood group. When this and this comes together, I get IA, IB. That means AB blood group. When this and this comes together, I get IA, IB again. That means AB blood group. When this and this comes together, I get IB, IB. That means what? B blood group. So how many phenotypes are possible? Physical nature. One is A blood group, one is AB blood group, and one is B blood group. So 3 of them. I will not count it twice, only once. Okay, if the blood groups are repeating, I will only count it once. So 1, 2, and 3. 3 phenotypes, physical regions are possible. What about genotype? 1 IA, IA, 1 IB, IB. Okay, 2 of them are done. And these are same IA and IB, so I'll count it as 3. 3 genotypes are also possible. Please understand this and do accordingly. Done? Done, ma'am. Okay, so let's go ahead with the next thing, the next exception which is there. Now this exception is given towards the end, it's like more beyond, it's not given over here, but I thought I should be explaining you, which is why I am here with this, and that is polygyny. Polygyny is also counted to be an exception. Why? Because Mendel, according to Mendel, he said that one gene which is there, it codes for one particular character, right? Now what if I tell you multiple genes can code for or basically affect the character which is there? Multiple genes which are there, they affect the character. And why I have shown you so many people's image with different skin tones. You can include me also over here. Okay, why do we all have different skin tones? Or why do we have different skin tones? You know the answer for that? If at all you do not know, then it's high time to know about it. Listen to me very carefully. There are certain characters, and this character is something that you need to remember. One is skin color and one is your height. They are polygenic characters. That means these characters are coded by multiple genes out there. For example, I say I have A, I have B, I have C. All of them, they contribute to my skin color. So over here the combination is AA, BB and CC. If I have a combination like this, I will be having the darkest skin tone. The darkest skin tone. Suppose I change something over here, there's a small allele, that is, just a second, small a, then now the genotype is capital A, small a, capital B, capital C. This is going to be lighter than this, the shade which was there. Now suppose you have small b over here. Now it is capital A, small a, capital B, small b and capital C. This is going to be lighter than this also. Which one is going to be the lightest skin tone? Which one do you think is going to give us the lightest skin tone when the condition is like this? All of them are recessive alleles. Small a, small b, small c. This is going to give me what? The lightest skin tone. The irony is that here, not just one gene is coding for a character, multiple genes, my dear children, are coding for a character, that is your skin color. And the lightest skin tone is observed when you have this. This is an important question that you have. Which one gives the lightest skin tone? You might be given options. You have to choose the recessive one. Your height is also like that. Both of them, they come under polygenic inheritance. So traits which are controlled by multiple genes are called as polygenic traits. Example, skin color and also human height. Each dominant allele can add to the trait. That means it can increase the color out there, while recessive allele reduces it. So more dominant alleles result in darker skin, while more recessive allele will be resulting in what? Lighter skin. That's all. That's all. That is polygenic inheritance. So first exception, what was what? Incomplete dominance. Second was multiple allelism. Third one was codominance. And the fourth one is polygenic inheritance. Now you have a fifth one also, and that is known as Pleiotropy. Again, you have heard that one gene which is there codes for one character. In the previous example, we saw that multiple genes are coding for one character. That was an exception. Here, one gene does not just code for one character, but can code for multiple characters out there. This is known as Pleiotropy. Do not get confused. There is no point of getting confusion also, because polygyny means multiple genes are there, right? Here it is Pleiotropy. So one gene is there, but this gene is controlling multiple character. The best example for this is a disease that is known as phenylketonuria. You might have heard about this, PKU, right? Phenylketonuria. What exactly happens over here? There is a gene that codes for a very important enzyme, that is phenylalanine hydroxylase. This gene is getting mutated. That means this enzyme is not going to be produced. If this enzyme is not going to be produced, what is going to happen? The phenylalanine, which is an amino acid which is there in our body, it is not going to convert into tyrosine. It will not convert into tyrosine. Okay, so what is going to happen? If this conversion does not happen, the phenylalanine accumulates in the body in the form of phenylpyruvic acid. What acid? Phenylpyruvic acid. And this phenylpyruvic acid accumulation can lead to what? Mental retardation. Can also lead to what? Mental retardation. Okay, fine. Got it? So that is one character that is being affected, right? No. So basically, this particular tyrosine that you see, when it is not being formed in your body, in your body, melanin is not going to be formed. If melanin is not going to be there, you will be having reduced pigmentation. That means you might have seen people with white patches out there, right? So reduced pigmentation will be there. Reduce pigmentation of what? Of your hair and also your skin. Some people just read it reduced hair. So it's not like hair is not getting reduced. Pigmentation is getting reduced. Why? Because the tyrosine is not there. Tyrosine is super important for formation of something else that is responsible for our skin color. Got it? So basically 2 characters being affected because 1 gene is getting mutated, right? This is an example of pleiotropy. So when a single gene influences different metabolic pathways or phenotypes. Phenylketonuria is an example caused by the mutation in phenylalanine hydroxylase gene. The phenylalanine hydroxylase enzyme converts phenylalanine into tyrosine, and its absence, phenylalanine accumulates to form phenylpyroic acid, causing what? Mental retardation. The second character it hampers is reduction in hair and skin pigmentation due to low tyrosine levels. I told you, reduction in hair, don't read like that. Reduction in hair and skin pigmentation, read like that. Okay, why? Because tyrosine level is also less. Done. This is your 5th exception. So these were the exceptions that you had for what? For Mendel's first law of inheritance, or basically not first law of inheritance, generally Mendel's study of monohybrid cross. Now, there is one more example given in your NCERT, and that is about what do you call the starch grain size and the shape. So this is an example both for incomplete dominance as well as pleiotropy. Listen to me. Why? Incomplete dominance, why? I'll tell you. So capital B, whenever it is there, it shows that there is a large starch grain. The starch grain will be large in shape. Now, large in size. What about the seeds? Seeds are going to be round also. If at all, there is capital B. If there is small b, it is going to be small starch grains which are there. Okay? And the seed shape, if at all you see, it is going to be wrinkled. Fine. So basically, one set of gene coding for 2 characters, which is why it is an example of pleiotropy. Now, why is it an example of incomplete dominance? Let's see that. Capital B, small b, when both of them come together, it will give rise to medium starch gains. So we had large, we had small. So when capital B, small b came together, we expected the large character to be shown because capital B is the dominant one, right? But over here, they are showing incomplete dominance, which is why you see medium-sized one. Okay, medium-sized starch gain. What about the seed shape? In seed shape, my dear children, it is still round because capital B is dominant out there. It is still round. So when you talk about shape, yes, capital B is dominant. Okay, when you talk about the size, capital B is incomplete dominant over the small b. So what does it show? For seed shape, capital B is dominant, but for starch grain size, it is incomplete dominance that you see. This is an exception given in your NCERT at the end. So some people ignore it, but we do not know, right, what questions could be coming at what point. So I did not want to take that chance. Please make sure you know about that also. Cool? Fine. Now we'll go ahead with Mendel's dihybrid cross experiment. And from this point onwards, we have to be a bit fast. Otherwise, this chapter will also take too long to get completed, right? So what is Mendel's dihybrid experiment? Over here, instead of talking about one character, you are going to talk about what? 2 characters, which is why it is known as what? A dihybrid cross. So what exactly happens up over here? In a dihybrid cross, my dear children, okay. So I am going to take 2 characters at once. For example, and this is something that confuses you guys. Just give me a second, someone is knocking the door. Okay, so over here, I am going to take 2 things, that is the shape and the color. Okay, so the shape is, suppose it is round and yellow. This is what? Seed shape and seed color. Similarly, over here I am taking green. Just a second. Wrinkled and green. I'm taking wrinkled and green. So round and yellow and wrinkled and green. Here also, this is what? The shape, and this is going to be what? The color. Agreed? Agreed, ma'am. Okay, fine. Now listen to me very, very carefully. Round which is there, it is represented like this, capital R, capital R. Yellow which is there, it is represented like this, capital Y, capital Y. That means they are homozygous, they are pure breed round and yellow. Both the alleles are same, right? Here the wrinkled one is represented by small r, and the green one is represented by small y. This is what, again, homozygous, and these are recessive traits. These are what? Dominant traits out there. We have studied in seed shape, my dear children, round is dominant and yellow is also dominant color, right? Now, how do you form gametes for this? This is difficult for you. So you have to use your imagination up over here. So suppose you have chromosome like this. Here you have the genes for what? The shape. Here you have the genes for what? The color. From this pair only one can come. Right? Only one, because that is how we studied last segregation. Meiosis is going to happen. Only half is going to come. So suppose it is capital R. Anyway, it is going to be capital R only. From here also only one can come. Whatever you do, it will be capital Y only. So in one thing, in one gamete, you are getting two chromosomes, but two different characters chromosomes you are getting. From this pair only one, from this pair only one. Similarly, the same thing happens over here. If at all you consider small r and small y, and small y. From here only one, that is small r. From here only one, that is small y. These are what? The gametes. Now we need to fuse them. How do we fuse them, my dear children? You cross this with this. And how do we fuse this? Listen, capital R and small r comes. Capital Y and small y comes. Capital R and small r comes. Capital Y and small y comes. How does it appear to you? It appears like this. So basically, one is capital R and the other one is small r. One is capital Y and the other one is going to be what? Small y. This is what you mean by this. So in the first generation that you see, every single plant which is there, it is going to show what? Round and yellow character. Round and yellow character. Okay, so that means only the dominant characters are being seen. Round and yellow is dominant character. That is the only thing that is being seen. But is it homozygous? No, Baba, it is heterozygous. Now we have two different alleles for two different characters out there. So Mendel thought, why not cross it? This is known as selfing. And then he went ahead with the selfing process. Again, how do we do that? So parent over here is going to be capital R, capital Y, small y. Capital R, capital Y, small y. Right? Now, the gametes which are there, you have to pay your attention over here. The gametes which are there, they are going to be like this. Capital R, capital Y, small y. Now, when you form the gametes, from this pair, one can come. Suppose I have taken capital R. From this pair, one can come. Suppose it is capital Y. This is one gamete. Another gamete, from here you got capital R, from here you got small y. This is another gamete. What about again? This is going to come small r, and from here capital Y can come. This is another gamete, and again the small r is there and you get small y. This is another gamete. So how many type of gametes are you getting over here? 4 type of gametes you get over here. Now from the same parent, this is also same type of thing. You get the same type of gametes. You put them here, and make the Punnett square. In the Punnett square, how are you going to do the calculation? It looks like this. So capital R came over here, capital Y came over here. Let's take a different example. This and this. Capital R, and capital Y, small y. You get it? So how many combinations are you going to get? You are going to get 16 combinations. Some of them are also going to be repeated. For example, if at all you see, this and this is repeated, right? There are so many combinations which are going to be repeated. So how many unique genotypes will you be able to see? How many unique genotypes? That is not being repeated, only once we are counting it. How many unique genotypes are you able to see over here? You will be able to see 9 unique genotypes. Always remember, out of 16 which are there, 9 of them are the unique genotypes which will be seen. Now what about the phenotypic ratio? What about genotypic ratio? Forget about it. Phenotypic ratio, can you tell the phenotypic ratio? It is going to be 9:3:3:1. What does 9 stand for? Phenotypic. What is a phenotype? Dominant phenotype? It is round and yellow. So let's make it round and yellow. The maximum of them will be having that phenotype. 1 means it's the least one. It is going to be the recessive parent that we took. What was the recessive parent that we took? Wrinkled and green. One of them is going to be wrinkled and green. Now 3 and 3 which are over here, they are the new characters which are there. So basically one is going to be round, and with yellow here, now this time you have to bring change, it will be what? Green. Similarly, over here also, you had wrinkled and green, you make it to wrinkled and yellow. Okay. This is how it goes. So basically, two characters that you see over here are non-parental. You do not see it in your, what do you call, in the parents. And my colleague is right over here smiling at me, making sure. So yes. So this is how it goes. So 9:3:3:1 ratio is there, and this is super important for you to know. This is what the phenotypic ratio. Okay, and also one very important thing, if at all you're given this question, okay, I will be showing you that question. Let me show you that question. But anyway, just remember 6 of them out of 16. So 9 plus 3 plus 3 plus 1 is equals to what? 16. So 6 of them, my dear children, are going to have what? Are going to have a different thing, non-parental character. And why is it happening? Why do you see non-parental character? Because of one very important thing, and that is the third law of independent assortment. It says that whenever gametes are being formed, a pair of alleles for one particular character are going to assort independently of the pair of alleles for another character. That means when capital R came from here, it didn't say that I'll also have capital Y. Sometimes it was with capital Y, sometimes it was with small y. So from a pair of alleles, one desegregated, this is law of segregation, right? Law of independent assortment means when a pair of two alleles are there, one pair of alleles are going to segregate independently of another pair of alleles which would be there. Until now we studied about only monohybrid cross, but now we are talking about dihybrid cross. Okay, and since you have these combinations out there, they are doing independent assortment. That is why you are getting different gametes. And when you get different gametes, you get non-parental characters as well. You got it? That is the basic thing out there. Now, so did Gregor Mendel find something unique? The answer is pretty much yes, the law of independent assortment. And what does this law say? When two pairs of traits are combined in a hybrid, when two pairs of traits are combined, segregation of one pair of character is independent of other pair of character. That is, segregation of the seed shape is different from segregation of the, the seed color. Fine? Yes. Now I want you guys to try it out. A pure breeding pea plant, that means, pure breeding means alleles are going to be same, with round and yellow seeds. It's like this. Pure breeding means it is going to be like this. Was crossed with pea plants having wrinkled and green seeds. Wrinkled and green means just like what we did now, right? On selfing of F1 hybrid of this cross, 64 progenies were obtained in F2. Find out the number of F2 progenies showing non-parental character. Showing what? Non-parental character. This is super important. So out of 64 progenies, how many of you do you think they are going to show the non-parental character? So options are given over here, A, B, C, D. Check it out. Go, fast. Try it out. Any idea? Listen. So we studied about 9:3:3:1 ratio, and we studied 6 of them out of 16. Okay, 6 out of 16 are what? Non-parental ones. Here you have to find it out of 64. So you basically do the 6 out of 16 into 64. When you do the calculation, 6 into 4 is equals to what? 24 of them. 24 out of 64 progenies, my dear children, are going to show the non-parental. How did you get the 6? 3 plus 3, 6. Out of what? 16. Now here it's out of what? 64. So basically you do this. This is the basic calculation which is there. This came in 2020 in KCET examination. Got it? Yes, ma'am. Fine. Now, I have a homework question for you which you have to do from your own end and put it down in the comment section. Listen, I generally check all the homeworks that I have given, and I really appreciate you trying it out. And if you know or not know, please let me know. If you're facing difficulty, let me know. I am a teacher. I am here for the same reason, to explain you guys. So there is no point in being shy and not commenting down the answer. You please attempt. That is more than enough. I'll remember your names when you attempt. No, I remember, okay, this was the person who watched it and commented. So in a dihybrid cross, the F1 plants, that is capital R, small r, capital Y, small y, are self-hybridized. The ratio of segregation of yellow and green in F2 is. The ratio of segregation of yellow and green in F2 is. Yellow and green. You are not being asked about both the characters, that is round and yellow. No. You're just asked about yellow and green. I'll give you a hint from here. Okay, from where? from here, spot the ones which had yellow and green. So 9 and 3 of them have yellow, wrinkled yellow, round yellow. And green is this. Now you can try to find the answer by yourself. I gave you a hint also. Okay, so that was the max I could do for you as of now. So we studied about the exception. We studied about the third law of Mendel also. Even though there are exceptions, yes, other things were correct. Most of the times it was correct, right? But you know what? One day, everything went wrong. Everyone was against Mendel. They said, and the reasons why it could have happened is that the communication was so difficult at that point of time. As of now, we have Snapchat, WhatsApp, Instagram, a lot of things to communicate. Hey, Chintu, what are you doing? Or whatever things you can do, right? But over here, it was not like that. Second thing, his idea of discrete factors and no blending was not accepted. As I told you, blending was not accepted. So he said that, so basically he said that units are there, capital A or capital T, small t, it will always give a tall. There is no medium character that was being seen, right? So this was not accepted. Using mathematics in biology was unusual and not accepted. So previously he was acclaimed for that, and now people were like, you're using mathematics in biology? Now it is not good. Fourth thing, he had no physical proof of what genes were and how did they look. Because, my dear children, he did not have microscopes being built by then, which is why he did not have any idea. Now you think about this genius man who without any much resources was able to find a lot of things, but all of them were against him, and that is the reason. So you have to write the reasons if at all you are asked, why was Mendel's work not being credited about. Okay, but don't worry, every time in our life we have downfall, but after that comes a twist. So if at all you are facing a downfall, remember there will be a twist in your life as well. And the twist came after 37 years in his case. 37 years. Okay, why do I say that? So there were 3 scientists, Carl Correns, Erin Voss-Schemark and Hugo de Vries. Okay, Correns, Schemark and Hugo de Vries, they rediscovered Mendel's work in the year 1900. Ma'am, is this an important question? Very important for your PU2 and your KCET. They always ask this. Correns, Schemark as well as what? Hugo de Vries. In the year 1900, rediscovered Mendel's work. Why? Because better microscopes were there, and by now they could reveal the structure of chromosomes. Why do you call it chromosomes? They are colored bodies. You can find it them. They can take up the stain, which is why you call them chromosomes. In the nucleus, which doubled and split during the cell division, they were able to find all of this thing, which is why they rediscovered Mendel's work in the year 1900. And not just that. Okay, this was Correns, Schemark as well as Hugo de Vries, right? After that, by 1902, that means after 2 years, 2 heroes also came into the picture. What was their name? Their name was, my dear children, Walter Sutton and Theodore Boveri. Walter Sutton and Theodore Boveri. What did they do? They came up with a very important thing that is known as chromosomal theory of inheritance. Chromosomal theory of inheritance. What was it all about? Listen to me carefully. They said that when you talk about the chromosomes, you know that when meiosis has to happen, the chromosomes, they split, and one chromosome goes up here, one chromosome goes up here, right? So they said that whenever chromosome was splitting, so were the genes which were being carried. Yes, so when this chromosomes were split, the gene also got split. And this is exactly what Mendel said. Mendel said, from a pair of factors, one is going to go into one gamete, right? Yes, so Walter Sutton and Theodore Boveri also had a similar thing, but they associated chromosome with it. He did not have, Mendel did not have any idea about chromosomes and things like that, right? But they came into the picture and they said, the movement of the chromosome is parallel to the movement of the factors that probably they carried. This was chromosomal theory of inheritance. So over here, I'll just tell you, behavior of chromosomes was parallel to the behavior of genes and used chromosome movements. This is what they said, but I'll tell more about it in here, that is in the chromosomal theory of inheritance. Sutton and Boveri argued that Pairing and separation of pair of chromosomes. Pairing and separation of pair of chromosomes would also lead to segregation of the pair of factors that we carried. So whenever a chromosome is being segregated, obviously it will also lead to the segregation of the factors that they are being carrying. So Sutton united the knowledge of chromosomal segregation with Mendelian principles and called this as chromosomal theory of inheritance. So even though we say that chromosomal theory of inheritance was given by Sutton and Boveri, in your NCERT one line is given, Sutton united the knowledge of chromosomal segregation as well as what the Mendelian principles and came up with chromosomal theory of inheritance. So my dear children, which year was in which year was Mendel's work rediscovered? 1900. By whom? Carl Correns and Schemark. And then you have Hugo de Vries. Okay, in which year did they give, which year did they give chromosomal theory of inheritance? Two people gave in the year 1902, just 2 years after that. It was Walter Sutton and Theodore Boveri. Now, obviously, you need to prove this down, right? Right? You need to prove all the things that you are basically claiming for. So that is why another guy came into the picture and his name was Thomas Hunt Morgan. His name was what? Thomas Hunt Morgan. And why did he love fruit flies? So basically fruit fly is the other name for Drosophila melanogaster. What is it? Drosophila melanogaster. And why did Thomas Hunt Morgan do his experiment on this particular organism? First of all, what was he experimenting? He was trying to give the experimental proof for chromosomal theory of inheritance, for which he used the fruit flies. And the reason why he used the fruit flies is that they grew on a simple medium. They did not need fancy medium. Today I want samosa, tomorrow I want jalebi. It's not like that, okay? So they were okay with growing on simple medium, and they had a very short life cycle. That means just up to 2 weeks. So lifespan was very, very short, which is why if at all there are changes, any changes, we could observe it very easily because of this shorter life cycle. Third thing, high progeny count. So whenever they mated, they gave rise to so many number of progenies, which was also a very good thing. Clear male and female distinctions. Male and female were clear distinction, and you should be knowing that females are larger in size than males over here. Females were larger in size than males over here. Now, visible heredity variations under low power microscope. You could also see their variations under low power microscope, and that was really easy for identifying a lot of things. So they were grown on a simple medium. They can have a short lifespan, that is just up to 2 weeks. High progeny count were there, clear male and female distinction was there, and visible hereditary variations were also visible over here. Done. Okay. Now, did he find something else? This is Thomas Hunt Morgan, by the way. Did he find something else? The answer is yes. He found 2 things, and that is what? Linkage and recombination. And I am going to explain you that over here. Listen to me carefully. If at all you listen to me carefully over here, your PU2 questions, easy. Your KCET, every case, MCQ based on this topic, also easy. So it goes like this. Suppose you have 2 chromosomes having alleles like this. You have another chromosome having alleles like this. So capital A, small a, capital B, small b. Capital A, small a, capital B, small b. The difference that you see over here is that the distance, right? The genes are far away from each other. Here the genes are very close to each other. First of all, whenever we say the genes are very close to each other, I'll call this as tightly linked genes. What do I call this? Tightly linked genes. When genes are very far away from each other, I'll call them as loosely linked genes. This feature is known as linkage. When they are close to each other, we call this as linkage. Okay, ma'am? Now suppose I am cutting off this portion from here. This portion goes this side and this portion goes that side. What is going to happen? Let's see. Small a, I'll just make the shape a bit big so that you understand it. Okay. Small a came this side, capital A went that side. Small b is that side only because only the lower part is being exchanged, and capital B remains on this side, right? What do you see now? you see a new combination being formed. Small a was with small b, but over here it is with capital A. Capital A was with capital B, but here small a is with capital B. What do you see this? What do you see? What do you call this? You call this as recombination, or basically what? A new combination out there. And isn't the recombination percentage very high? There is a new combination being formed, right? When they are very far from each other. So I'll say the recombination is very high. If at all you didn't understand it still, don't worry. You will understand it now. It's the same thing I repeat over here. I cut these portions from here. What am I going to get? Capital A, small b was on this side. Now, sorry, small a, small b was on this side. Now, here what you will be having? Capital A, capital B. Here what will you be having? You have small a, small b. But if at all you see, it's together. They are also together. They were together. They are together now. Recombination is happening? Yes. Recombination is definitely happening. It's not like new combination is not being formed, because the upper gene is different, right? So with that, if you compare, yes, recombination is happening. But the percentage of recombination which is there, my dear children, it is going to be less. Here, it is going to be more. Total structure is entirely being changed somehow, right? So what can we conclude from this? When there are tightly linked genes, you will be having lesser recombination. When the genes are loosely linked, you will be having what? More recombination. This is what was told by T.H. Morgan. And he basically did his experiment with the help of, as I told you, the characters of the fruit fly. So he came up with a conclusion, and that is this. Listen to me carefully. So there was a gene coding for yellow body, and there is a gene that is coding for white eye. Yellow body, and here it is white eye. These two genes are very close to each other. That means whatever you do, their recombination is going to be less. Now over here you have another gene, and that is for miniature wing size. That is for your wing size. If at all you talk about this and this, now they are far away from each other. These two genes are going to be far from each other, which is why here the recombination is going to be high. You get it? Here, since they were close to each other, these two genes, yellow body and white eye genes, are going to show lesser recombination. Why? Because they are close to each other. Whereas when you talk about white eye and miniature wing, or basically yellow body and miniature wing, they are very far from each other, which is why there are chances of higher recombination to happen. Because in the case of sexual reproduction, my dear children, this is very common. This is known as crossing over. This is known as crossing over. So whenever crossing over happens, there are chances for recombination. Now you have to just identify whether it's more or less. So what is the maximum percentage of recombination? It is 50%. Maximum percentage is 50%. Over here, this and this character showed almost 37.3% recombination. That is very much close to 50%. What about these two genes? Here it is very less. It is almost 1.3% recombination out there. That is very less recombination. So you should know this. You can get questions on the basis of this. Why did they show lesser recombination? Because they were tightly linked genes. Why did they show more recombination? Which was because they were loosely linked genes. You got it? Got it? Recombination happens due to what? Crossing over. Fine, that's all. This is what you need to know from this particular thing. Now I have already discussed it. What is linkage? Physical association of genes on the chromosome is known as linkage. It can be tightly linked, it can be loosely linked. And one more example, when you and your best friend are there and you're very close to each other, do you think any other friend can be formed? No. So you say less recombination. When you and your friend are distant from each other, you're not that close, what is the probability that someone else can also come into your group? So high recombinations. Okay, you can remember like that. Now what is recombination? Generation of non-parental combination, which was not seen in the parent. That is what you mean by recombination. And this is a result of what? Crossing over. So according to Mendel, also new character, non-parental character was seen. But that was because of what? Independent assortment. The genes were independently assorting. Here, for T.H. Morgan also, you find non-parental character. Why was it happening? Because of crossing over. And crossing over was leading to recombinations. Okay, please know the difference of that also. Fine. Now, as I already told you, that is this. Listen to me very carefully. Example, white and yellow genes, white eye and yellow body, 1.3% recombination. That means very less. Okay, they are tightly linked. 37.2 for white and miniature wings. 37.2 for white and miniature wings. Or not 37.3, it was 37.2. Why? Because they are far away from each other. That's all. So if at all you see this and get shocked, don't worry. Just read this thing and just study what I have taught you. That's all. That's enough. So there's something that happened that shocked the teacher. Why? Because the student rocked on the other side. Who was the student? His name was Alfred Sturtevant. His name was Alfred Sturtevant. And why did he shock everyone? Because, my dear children, yes, this guy gave the linkage and recombination, but with that piece of information, Alfred Sturtevant came up with one very important term, and that is known as gene mapping. What do you mean by gene mapping? He said that when you say that recombination percentage is more, I just understand the gene for this is very far from each other. When you say that recombination is more, that means they are very far from each other. And with this percentage, I am able to identify their location. I am able to map these genes. So that is known as gene mapping, and that was given by Alfred Sturtevant. Please remember, this is a very important thing out there. Got it? Yes, ma'am. Got it. Okay, since you got it, let's move ahead. So I already told you, I already told you, he discovered what? Gene mapping. Okay. Now, the sex determination is the next topic that we have. An odd topic, okay, over here, but very interesting. So we are going to study it. Just give me 1 second. Okay, I'll come, drink some water, and I'll be back. Okay, so let's go ahead with sex determination, and here it goes. Okay, so now we are back again with the story of Hen King. Well, my dear children, he found out one very important thing. That is, basically, in organisms like grasshopper, he saw that, 50% of the sperms were carrying chromosomes, and 50% of the sperms were not carrying any X chromosome, that is 0 sex chromosome out there. He was like, oh my God, how's that possible? So basically, why do you think that was happening? So listen to me carefully. This is a thing with the insects, and for example, I have grasshopper baba. Grasshopper has XX chromosomes. That means if at all there's a combination like this, it is going to be a female. Grasshoppers also have male grasshoppers where the chromosome combination is XO. And by that, I mean either it can be X chromosome that is getting passed, or either no chromosome that is going to get passed. So when you make the gametes, you get something like this. A gamete having nothing. A gamete having X chromosome. Two gametes having X chromosome. When this and this comes together, you get XX. That means what? A female. When this and this comes together, you get XO. That means one X chromosome is only there. It cannot be a female. It has to be a male. Again, when this and this comes together, you get XX. Again, a female. When this and this comes together, you get XO. Again, what? A male. But if at all you see, in the case of male grasshoppers, 50% of them are going to carry X chromosomes, and 50% of them are going to carry no chromosomes. That is what Hen King said. 50% carrying X chromosome, 50% not carrying the X chromosomes. Right? Got it? This is the type of sex determination that you see in grasshopper. And what do you call this type of sex determination? You call this as XO type of sex determination. And here you see one very important thing. Females produce the same type of gametes, X and X. All of them carry the same chromosome, X and X. But males, if at all you see, they produce two types of gametes. One that carries X chromosome, one that does not carry anything. So I can call this condition as heterogamety. What do I call this? Just a second. I call this as heterogamety. But who is producing different type of gametes? Male. So I'll call this as male heterogamety. Hetero means two. Different. Okay, so male heterogamety is an example. Grasshopper is an example for male heterogamety. Apart from that, do we have any other examples for male heterogamety? The answer is yes. For example, if at all you talk about human beings, and if at all you talk about Drosophila, what do you see? Drosophila and human beings, my dear children, we have XX, XY type of sex determination, where XX is again going to be the female and XY is going to be the male. Here, females produce two type of gametes, same type of gametes, that is XX. Male produce two type of gametes, that is X and also Y. When this and this comes together, you get XX, that means it's a female. When this and this comes together, you get XY, that means it is a male. When this and this, again a female. When this and this, again a male. So basically, can I say that 50% of them are going to be females and 50% of them are going to be males? Yes, ma'am. How is this an example of male heterogamety? The reason is that females do not produce two type of gametes. Males are the ones that are going to produce two type of gametes, half of them carrying X and half of them carrying Y chromosomes. So for male heterogamety, you have three examples: human being and Drosophila showing XX, XY type of sex determination, and grasshopper that shows XO type of sex determination. Done, ma'am. Okay, when it is done, we have to go with the next topic. What is the next topic? Female heterogamety. I hope this is clear, right? Female heterogamety. Have you seen any organism where females produce two type of gametes? If at all you have not seen, it's time to see. Is the case of birds. In the case of birds, my dear children, what is going to happen? In the case of birds, you have, just a second. You have ZZ and ZW type of sex determination. What is ZZ? It is going to be female, no ma'am? No, it is going to be male over here. I told you in case of birds, you are going to see female heterogamety. So that means this is the female, ma'am? The answer is yes. Here you are going to get two gametes, one carrying Z, another carrying W. Here you are going to get the same type of gametes, that is ZZ. When this and this comes together, you get ZZ, it will be a male. When this and this comes together, you get ZW, it is going to be a female. This and this, ZZ, that is again a male. This and this, ZW, and that is a female. How do you remember it easily? W for women, and that is for ZW. ZW means it is going to be female, and this is the only example according to your NCERT where they show female heterogamety. Rest all of them are male heterogamety. Okay, so grasshopper and the other insect we saw XO type of sex determination. In the case of human beings and Drosophila, XY type of sex determination. In the case of birds, ZW type of sex determination. Now there is one more organism out there which is going to shock you, and that organism is what? Honeybee. Honeybee. Okay, in honeybee, dear, they have haplo-diploid type of sex determination. Haplo-diploid type of sex determination. What do you mean by that? Okay, I'll tell you. First of all, understand the basics. Diploid means 2n condition. Haploid means half of it, that is n. Suppose we say we have 46 number of chromosomes generally. In our gametes, we have only half of it. That is what? 23. Right? Right. So over here, in the case of honeybees, they have 32 number of chromosomes in their diploid condition and half of it, that is 16, in the haploid condition. If at all an organism is diploid in nature, it is going to be a female. If at all an organism is haploid in nature, it is going to be male. This is known as haplo-diploid sex determination. Didn't understand? Listen to me carefully. Suppose there is a female, and this female has what? 32 number of chromosome. There is a male, and as I told you, the male will be having only 16, half of it, 16 number of chromosome. The female, when it has to form gametes, it will be undergoing a process that is known as meiosis. Meiosis is a reductional division, which is why in the gametes you are going to have only half the number of chromosome, that is 16. Right? Male already has 16, so to form gametes, it cannot undergo meiosis because it will be half then. It rather undergoes equational division, which is known as mitosis. So here also the gamete has 16. Okay, here it undergoes mitosis, here it undergoes meiosis. Now suppose the egg and the sperm fuses up over here. 16 plus 16, you get 32. Suppose this egg which is there, it is not fusing. It is not fusing with anything. It is not undergoing fertilization. Still it forms something. Can you guess what is going to form? And what is this? 32. Haploid, diploid. If it is diploid, it is definitely going to be a female. What about this? Here, it has not underwent any fertilization, and without fertilization also, it is able to give rise to a sex. That is what? Male. This is known as parthenogenesis. There is no fertilization, and since there is no fertilization, they will only have the number of chromosome, that is 16. Now, if at all I ask you, does it have a male parent? No. It just has a female parent. Does it have a grandparent? Yes. Why grandparent? Because female to come, you need the contribution from a male N and a female N, right? So N, then only it will be coming up right there. You get it? So it does have a grandparent. It does have a grandfather. But there is no father. But there is no father out there. This is also given in your NCERT. This is haplo-diploid sex determination, which you see in honeybees. Females are going to be diploid and males are going to be haploid. The process where you convert or basically form the males without fertilization is known as parthenogenesis. This is different from parthenocarpy. Yes. Okay. Got it? Yes. So if at all we study all of it now, I told you XO type of determination is seen in grasshopper, right? Sex determination, male heterogamety, female heterogamety. For XY, it is Drosophila and human beings. XO, it is grasshopper. Female heterogamety, you only see in birds. Example is DW. Okay, haplo-diploid sex determination, I told you it is there in honeybees, and this is what you see. Now, over here, decoding the twister in your NCERT. They do not have father, thus cannot have sons, but have grandfather and can have grandsons. So, since there is no father, we cannot say there is son. Also, we say there is grandfather and grandson relationship happening up there in the case of honeybees. Okay, do not write parthenocarpy and come. I am telling you again. Okay. So, parthenocarpy is seen in the case of flowers. That is different. Okay, now we come up. Parthenocarpy is what? Formation of a fruit without fertilization. That is parthenocarpy. This is parthenogenesis. Now we come up with one very important thing, and that is mutation. What do you mean by mutation? If at all the DNA is changing, the DNA sequences are being changed, and that causes some problems in the phenotypic expression or the genotypic expression, we call this as what? Mutation. So mutation is a phenomenon that alters DNA sequence leading to change in the genotype and the phenotype. Now, is it good or bad? The answer is both. It can be good because it can lead to evolution. Some mutations are bad, it can lead to diseases out there. For example, there is an example given in your NCERT, and that is about point mutation. Point mutation means, also you need to understand, listen, we have our DNA. The DNA which is there, one strand of it, it is getting transcribed to what? RNA. What type of RNA? mRNA. And this mRNA gives the information to form what? Proteins. So if at all there is some problem over here, it is going to be reflected over here, and it will be reflected over here in the formation of protein also. So you need to understand that. That's a basic thing, right? So I am telling you there is a problem. The problem is that in the mRNA that you have codons, 3 triplet codons out there. On the 6th position, my dear children, on the 6th position, you should have had GAG codon, triplet codon. But here, in the 6th position, you have GUG. So A which was there, it is now changed to U. This codons which are there, they code for amino acid, and here it was coding for an amino acid, that is glutamic acid. But now, since you have a different combination altogether, it is going to code for another amino acid that is known as valine. The amino acids which are being coded over here, they come together and they form what? A protein. This is a normal protein. But here, since this amino acid is changed, do you think you get a normal protein out there? No, you get a different protein altogether. So this mutation which is happening only at a single base pair, you call this as what? Point mutation, change in a single base pair. And this is what exactly happens in the case of sickle cell anemia. You might have heard about this disease, right? Sickle cell anemia. In this particular disease, you have the change happening out there. This is super important for you to know. Okay, frame shift mutation is another type of mutation where what is going to happen? Suppose I have red cap and blah blah blah. There are three codons out there. In between that, if at all I am inserting a codon A, what is going to happen? My reading frame is going to be changed. Every time we just read three codons, right? So red and then ACA is going to be red. That means different amino acid is going to be formed. This leads to frame shift mutation. Or if at all you insert two of the nucleotides out there, still it is going to bring changes. What if I insert three of them? What if I insert three of them? So red, cap, and in between I have three diseases, red or cap. So basically three of the nucleotides, when they are put, the frames are not going to shift. It is still going to produce something, and here this particular codon is also going to produce something. That means it is also going to code for amino acid. Sometimes these three codons which are there, it can be a stop codon. It can stop the process of the whole translation as well. So that is a problem. If at all you are inserting three codons, this does not lead to frame shift out there. It does not lead to frame shift, but it can lead to stoppage of translation. Why? It can be a stop codon as well. If at all you are putting two or one codon in between, it leads to frame shift mutation. Or basically if you insert or you can delete also. If you're inserting one or two codons or you're deleting one or two codons, that also lead to what? Frame shift mutation happening. If you're inserting three codons, if at all they're not stop codons, it will go ahead, it will produce the amino acid, but if it is stop codon, it will stop the process of translation as well. Now, the things that causes mutations, what do you call them? You call them as mutagens. What do you call them? You call them as mutagens. So sickle cell anemia, this. Always remember A comes before U. So A was first, it is replaced by U. G comes before V, so it was glutamic acid first, now it is replaced by valine. Where is it happening? At the 6th position of the codon. Got it? And what is the protein being made? It is beta globin chain. So hemoglobin is, it's a very important protein. It is made up of two things, alpha globin chain and beta globin chain. So the protein that makes the beta globin chain is being affected, which is why hemoglobin also is going to be affected indirectly. That's all. Yes. So now we go about pedigree analysis on all the diseases which are there. So what exactly is pedigree? Analysis of traits in several of generations of a family is known as pedigree analysis. And over here, this particular picture is given, which is very, very important for you. You can get question, what does this mean? This means it's a male. What does this mean? This means it's affected male out there. What does this mean? This is female. What does this mean? This is affected female. What does this mean? This means sex is not specified. It can be male or female. What does this mean? That this particular person who is either a male or a female is being affected. So whenever you shade it up, that means it is affected individual. When there is a square and a circle and you show a line in between, this shows that the male and the female is mating. Now when you have two lines in middle, this is super important. When you have two lines in middle, that means what? It is mating between relatives. They are very close to each other, which is why you have two lines. You can remember like that. You have a separate term for it. It is also known as consanguineous mating. Okay, it is also known as consanguineous mating. Okay, now male and female crossed, that is mating happened. Now you get female and male child. General thing. Male and female crossed, and now you get a male child who is being affected. If at all you get this, and here it is written 5. That means 5 of children are being born. We do not know whether it's male or female. If it is colored, it means that 5 of the children are being born, and we do not know if it's male or female, but they are affected with some disease out there. Got it? This is the most important of all of them, consanguineous mating or mating between relatives. Now, I will be talking about the pedigree chart, but before that, I have to explain you the disorders. So we'll go with disorders first, and then we'll see pedigree chart. Okay? So you have two types of disorders altogether. One is Mendelian and one is chromosomal. Mendelian basically means, suppose over here, in the chromosome, I have a gene, and this gene is getting mutated, which means that protein is not going to be formed properly, and which is why I will have some disease out there. These type of disorders where the genes which are there on the chromosomes, they are being altered, and which is why you have a disease, you call them as Mendelian disorders. What is chromosomal disorders? By chromosomal disorders, I mean, suppose we should be having a pair of chromosome. Now I have an extra chromosome out there. That means some problem is going to occur with me. The number of chromosome is being altered. Similarly, sometimes I should be having a chromosome like this, but half of it is not there. Structural effects in the chromosome also lead to certain diseases. So these are known as chromosomal disorders. This is more related to genetic level, and this is what the chromosomal level. So determined by alteration or mutation in the single gene are there. Chromosomal disorders are the one where either the structure or the number of chromosomes are being altered. Right? So first we are going to go ahead with Mendelian disorders, and the first Mendelian disorder that we have is color blindness. And remember, in your NCERT, the ones which are discussed are all of them are recessive disorders. What do you mean by recessive disorders? You will come to know. And first two of them are sex-linked recessive disorders. What do you mean by that? Ma'am, I do not understand. Listen to me carefully. So color blindness and hemophilia are sex-linked recessive disorders. That means that it will only show up only if at all they are in recessive condition. Both the alleles are same. Okay. And also one more thing. Here the sex chromosomes genes which are there on the sex chromosomes, they are being affected, which is why you call this sex-linked recessive disorder. For example, if I say this is a female, right, and this is going to be a male, right? For a female to have a disease, she should have both of her chromosomes being affected, because only in recessive condition the recessive disorders are going to express. If at all she has one chromosome which is normal and other chromosome which is affected, still you have one chromosome which will dominate over this, and it's not a recessive condition, which is why she will not have the disease. She is going to be a carrier for the disease. Okay, for her to be diseased, both the chromosomes should be affected. For her not to be diseased, one chromosome, if at all it is affected, she can be a carrier. Or if at all there is no problem, she can be a normal person also. In the case of males, he does not have two X chromosome, only one X chromosome is there, and if this one X chromosome is affected, the person will be definitely having a disease. There is no such term like carrier males over here. If the male's X chromosome is affected, he will be having the disease, since there is no other X chromosome to overshadow this effect. That is why in sex-linked recessive disorders, it is males who are generally affected more. They are the ones who are affected more. Females sometimes, most of the times, can be a carrier for this particular disease. What happens in color blindness? People can't see any colors? No. It depends. There are different types of color blindness. In your NCERT, color blindness, the red and green color blindness has been mentioned. So over here, there is defect in the red or the green cone cells, leading to difficulty distinguishing what red and green color out there. And it occurs in about 8% of males and only about 0.4% of females. This data is super important for you to remember. 8% males and not 0.8, it's 0.4% females that are going to be affected. That means clearly it is mentioned that males are affected more. Now females are usually carriers unless they inherit two defective copies. Yeah, they are usually carriers unless they inherit what? Two defective copies from their parents. Probability, a carrier mother has 50% chance. Now, from this point onwards, everything is going to be literally real confusing for you guys, which is why I do not want that you get confused. I'll explain to it one by one, and you please listen to me carefully. Okay, it goes like this. First condition, where, just a second, the male is normal and the female, dear, she is just a carrier. Let's see, if she is a carrier, what is going to happen? This and this chromosome comes together, you get XX. That means a female who is normal. So I'll just write NF. Okay, going ahead. This and this comes together, you get XC. Here again, it's a female and she has one X chromosome which is affected. This female is going to be a carrier. She will not have the disease, she is just a carrier. Okay, this and this comes together, I get a male who is perfectly normal, so NM. This and this comes together, I get X and Y, and here the X chromosome is affected. So I get what? A male who is affected. This person is going to have the disease. So out of two males that you have getting, one male is definitely going to be affected. So can I say 50% of the males are going to be affected? There are two males, out of them one is affected. That means 50% of males are going to be affected. So carrier mother has a 50% chance of passing it to her son. And what about the daughters? A daughter will be color. Okay, we'll come to that. So I hope this is clear. 50% chance in the case of males. You're not talking about everyone together. Okay, if you're talking about everyone together, then out of 4, one of them is being affected. So 1 by 4 is equals to what? 25% of the children will be affected with the disease. 25% of the children will be affected with the disease. Got it? Similarly, 25% of females can be So you say 50% of the females can be carriers. 50% of the females can be carrier. Out of 2 females, 1 can be a carrier also. So 25% of the total offspring, 1 person can be affected, and that has to be a male. That will be a male. Okay, this is one condition. Now, going ahead. Again, the boy is normal. The female is having the disease. She is colorblind. And he is normal. Let's see. This and this goes. The condition is like this. She is going to be a carrier female. This and this goes. Again, she is going to be a carrier female. This and this, a male who is being affected. This and this, again a male who is being affected. So all the sons who are going to be there. So female, if she is affected, all her sons are definitely going to be affected, because in the sons you get only 1 X chromosome, right? And that X chromosome anyway is going to be affected. You get it? Do not even need to do the calculation. So all the males are going to be affected. 100% of the males are going to be affected if the female is having the disease. Whereas the females, my dear children, they are going to be just carriers. Okay? If the father is normal. Now, you only think, if you want to make the females being affected, you need 1 X chromosome from the mother and 1 X chromosome from the father. So at least, okay, let's see that condition now. Here, this time, the male is affected and the female is a carrier. Female a carrier and the male is affected. So what is going to happen? This and this comes together, you get a carrier female. This and this comes together, you get a female who is going to have the disease. This happens only when the father is having the disease and mother is either a carrier or will be having the disease. If at all she has a disease, then what is going to happen? All the females are going to be disease only, because from here you get XC and from here also you get XC. You get it? Got it? This is super important. Now, let's talk about the males then. This and this XY, this is going to be a normal male. This and this XCY, this is going to be a male who is having the disease. Okay. But anyway, males are being affected, whatever you do, right? So that is the thing. So for a female to have the disease, my dear children, at least the mother should be a carrier. At least the mother should be a carrier, and male should definitely have the disease. Then only you can transfer it to the daughters out there. This is what you have. So a daughter will be colorblind only if her father is affected and mother is at least a carrier. At least a carrier. She can be diseased also. Fine. This is all about color blindness that I had to tell you. Now I told you hemophilia, one more disease is there that is also sex-linked. It has the same pattern. So instead of C, you put H over here. Now, I want to tell you something about hemophilia which is different. What is that something about hemophilia which is different? So listen to me carefully. Hemophilia is a disease which is caused, if at all, a gene that is coding for a protein, and this protein is super important for clotting of blood, is being affected. So this protein is helpful for the clotting of blood and that is being affected, which means that if at all I get a cut and if I have hemophilia, the blood is going to ooze up and literally I will be dying at one point, right? It's a fatal disease that is there. Color blindness, you can live with it. But this is a fatal disease that I am talking about, which is why females, yes, she can be a carrier, she will not be having the problem. She can be just a carrier. But, my dear children, males on the other hand, if at all they have the disease, they will not survive until reproduction phase. They will not survive until reproduction phase. Females, they have a chance to be carrier. If at all females are affected, they will also die. But if at all the female is a carrier, she does not have any problem. Males, can I expect him to live until the reproductive age? No. Which is why this was the only case where the females were getting affected, daughters were getting affected. Which is why the chances of daughters being affected is very less. And close to nil. I'll say it's close to nil. because they cannot survive until that time. And if at all you're not getting one affected X chromosome, how are you going to make a affected female, right? So here again the males are being affected, and it's actually true that the females are not going to be possibly be affected because the fathers cannot be viable until that particular point of time in the case of hemophilia. So that is the difference that you have between color blindness and hemophilia. So I have a question for that, but okay, so that is this. So did Queen Victoria had hemophilia? Yes, she was a carrier. She was a carrier for this. So a single protein that is a part of cascade of proteins involved in clotting of blood is affected. Here a simple cut will result in what? Non-stop bleeding. Okay, the possibility of a female becoming a hemophilic is extremely rare. Extremely rare. Why? The mother of such female has to be at least a carrier and the father should be hemophilic. Usually they are unviable in the later stages of life, which is why this is not clearly possible. So it is very, very rare for the female to be affected in this particular case. Then in color blindness, yes, there were chances. Yeah, but here almost no. So this is the second sex-linked recessive disorder that you had. Two of them, color blindness and hemophilia, right? Now a question for you. A gene for hemophilia is located on X chromosome, hence it is normally impossible for what? Genes for hemophilia is located on X chromosome, agreed? Yes. Hence it is normally impossible for what? Hemophilic father to pass the gene to his daughter, carrier mother to pass the genes to her daughter, carrier mother to pass the genes to her son, hemophilic father to pass the genes to his son. What do you think? It's normally impossible. So ma'am, you told hemophilic father to pass the genes to his daughter, that cannot be possible. I said it's close to nil. I didn't say it's not possible. It is possible. If that happens, the child will be having the disease, right? So it is possible, but it rarely happens. If at all they survive until that particular point of time. So this is not wrong. Carrier mother to pass the gene to her daughter. Carrier mother can pass her gene to the daughter. Daughter can have at least one gene up there. This can be normal gene or this can be affected also from the father that it gets. But she can pass the normal. She can pass what? The gene out there. So this is also correct. Carrier mother to pass the gene to her son. She can also pass because son will also have one X chromosome and one Y chromosome from the father. So this X chromosome comes from the mother. So yes, he can also have it. So this is also correct. Hemophilic father to pass the gene to his son. Is that true? Hemophilic father, that means the father is like this. Can he pass the gene to his son? No. To his son, only the Y chromosome is going to go, not the X chromosome. So it is not merely impossible to do that. You get it? Hemophilic father cannot pass to his son. The mother can pass it. The father cannot pass it. Which is why you do not see male-to-male transmission. Okay, you are not going to see male-to-male transmission over here unless and until the female is affected. Okay. Now, so we discussed about that. Now we have the autosomal recessive disorders out there. I told you all the diseases which are there, they are recessive only. This is autosomal recessive disorder, and the first one is sickle cell anemia. So as I said about sickle cell anemia, this is super simple. Here, you need to understand that hemoglobin is there. Hemoglobin is there. Should have written it at a nice place, right? So I write it somewhere here. Hemoglobin which is there, it is made up of two things, alpha globin chain and beta globin chain. Here you are forgetting about alpha globin chain because you do not need it. In sickle cell anemia, it's the beta globin chain which is going to be affected, and that went. Okay, my God. You know, you need high patience to deal with things like this. So I was in here, hemoglobin, you had alpha globin, you had beta globin. Here alpha globin, you forget about it. Beta globin chain is going to be affected. The genes for formation of beta globin chain is located on what chromosome? Chromosome number 11. 11, chromosome number 11, okay? And if at all I am saying that this particular gene which is there, it has some problem. It has some problem, as in some mutation. What do I mean by that? I mean that in my mRNA, it was initially GAG as I said, but now it is what? GUG. Which is this codon? 6th codon. So initially what was I coding for? Glutamic acid. And now what is it coding for? Valine. It is now coding for valine. That means all the amino acids, when they come together, you have valine instead of glutamic acid now. Which is why the beta globin is going to be altered. If the beta globin is altered, that means the hemoglobin is altered. If the hemoglobin is altered, do you think it will be able to carry enough amount of oxygen? No, Baba. And not just that, this altered hemoglobin will change the structure of the cell. So usually the RBC structure is bioconcave shape, or you call it discoid structure, but now it turned into what? Sickle-celled shape. Sickle-celled shape. And when you have a shape like this, there are chances that it can cause clots. It can lead to what? Clots. I hope this is clear. So A was there first and then came G. So A in alphabet first, then comes U, not G, U, sorry. So A was there first and then came U. Here it was glutamic acid and now it is valine. Here also G comes first and then comes V. So this is the thing that is happening. Now, you need to understand one very, very important thing. That is, how is it transmitted? As I told you, it's a recessive disorder. So the normal gene which is there can be like this. This is a normal one, okay? Suppose both the alleles which are over here, they are normal, okay? Here also they are normal. From one parent, another parent, both of parents are giving normal genes. Whatever you do, the combination would be like this only. That means you do not have any problem. Now let's talk about a person who is a carrier, okay? So a normal gene is there, but an affected gene is there. Okay, this gene has been affected, suppose on chromosome 11. Similarly, you have another parent. He or she is also a carrier. Agreed? Now if at all you cross, let's see what you get. When you cross this and this, you get HBA, normal. When you cross this with this, you get HBA, HBS, going to be a carrier. When you cross this with this, you got HBA and HBS, again a carrier. So now you have got a normal person, two carrier people. When you cross this with this, what do you get? You get HBS, HBS. This person is going to have the disease. Ma'am, is it a male or female? It can be anyone. So can I say out of 4 offspring being produced, one of them is definitely going to have the disease if both the parents are carriers? Yes. So that means 25% of them are going to be affected. So if at all you get questions like this, when both the parents are carrier, how much of them will be having sickle cell anemia? 25% of them, 1/4. How many of them are going to be carriers? 2 out of 4. That means 1/2, 50% of them are going to be carriers. You get it? And the people who are having sickle cell anemia, do you think they can come mate and produce? No. That is definitely not going to happen, because again, there are less chances for them to survive up there. Okay, there are less chances for them to survive. So usually it's the carriers. And again, it can be male and female. This is autosomal disorder. That means it is not on your sex chromosomes. On the other chromosomes that you have, the genes of the other chromosomes are being affected. So always remember that also. So over here, let's read about it and get a better clarity. Caused by mutation in the beta globin gene, glutamic acid is replaced by valine in the position number 6 due to GAG to GUG substitution, GAG to GUG. Okay. Now, mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC. This mutant hemoglobin leads to what? Leads to different structural changes, polymerization, leading to what? A change in the shape of the RBC also. Because oxygen is very, very less. This hemoglobin is not able to carry oxygen, which is why all of this is happening. Now, from bioconcave disc to elongated sickle-like structure is going to form, which is again going to reduce the oxygen transport and also blood flow obstruction. Why? Because it is sickle cell shaped. So oxygen transport is going to be less and clotting can also happen. HBS, homozygous condition, that means a disease individual is being formed. HBS, that means heterozygous, they are going to be just carrier. And HBA, that is a normal beta globin gene that is going to be produced. If both the parents are carrier, the child will also have it. How much percentage of children, my dear children? 25% of them. 25% of them are going to be affected over here. I hope that is clear. And also remember, your mRNA is being affected. GAG to GUG, right? That means sometimes the problem can be here. So problem can be happening up here, why transcription was there, where RNA is being formed. So problem can be happening in the DNA also. So suppose in the DNA, if it had to be GAG, in the DNA it would be what? CTC. It should be CTC. But here in the DNA, there is some mutation, which is why you need GUG, right? For GUG, the opposite of it will be what? CAC. So your DNA could have been like this, which is why it is coding for something like this. We get it? If the DNA was normal, it would be coding something like this. So that can also be a reason. Either a problem directly in your RNA or probably a problem that was in your DNA and that is transmitted. Okay, so please know about that also. Have a clarity about that also. Okay, now going ahead. The second autosome linked disorder that is there, it is phenylketonuria. We already discussed about it. I don't think we need to discuss. What happens in phenylketonuria? The gene that codes for phenylalanine hydroxylase enzyme is being affected, which is why the conversion of phenylalanine to tyrosine does not happen. And when this conversion does not happen, phenylalanine is going to accumulate in the form of what? Phenylpyruvic acid. And phenylpyruvic acid, when it is accumulated, it leads to what? Mental retardation. Okay, and also, you know what, these compounds which are the phenylpyruvic acid, it cannot be reabsorbed by your kidney because and that is why it moves down from your body, and that gives a very bad smell in the urine also. And the color of the urine also is different. So these compounds are excreted in the urine due to poor kidney absorption. Please know about that as well. Okay, it's an example of pleiotropy. So I explained this, which is why I'm not explaining again. This is an autosome linked recessive disorder. So sickle cell anemia was a blood disorder, and also remember one more very important thing. Here in sickle cell anemia, I call this as qualitative disorder. Qualitative disorder. Why qualitative? Because here the quality of quality of hemoglobin is being compromised. Not the quantity, it is the quality that is being compromised. Now we are going to study about one more disease where it is going to be quantitative blood disorder, and that is thalassemia. Do you know, Amitabh Bachchan is a carrier of thalassemia? Yes, and a lot of us might be, which is why we all should be getting ourselves checked. You know, we should be getting it checked. So later on, when you grow up and you will be having babies, make sure you check it up. Otherwise, the baby might also have the disease. 25% chances there, right? So yes, listen to me carefully. Why does thalassemia happens? Again, it's a mutation that happens in the genes on their autosomes. Again, it's hemoglobin molecule, and this time it can be your alpha chain. It can also be your beta chain that is going to be affected. Alpha globin genes are there on chromosome number what? You tell me. Beta globin chain are chromosome number 11. This we studied. This is there on chromosome number 16. Chromosome number 16, alpha globin chain. Okay? And it looks something like this. I'll tell you. So, for alpha globin gene, my dear children, you have two genes. That means 4 alleles in total. One gene having 2 alleles, another gene having 2 alleles. Suppose one of them is getting mutated. One of them is going to get mutated. That will be a problem. Less amount of alpha globin gene is going to be produced. But if 2 of them are going to be mutated, which bit more lesser amount of alpha globin gene is being produced, still you have 2 of them, which is why it produces, but amount is getting less. What if 3 of them are affected? Bit more less. What if all of them are affected? No alpha globin chain. No alpha globin chain. And without alpha globin chain, do you think you can form hemoglobin? No. So if it is less, that is also a problem. If it is not at all there, that is a very big problem. You cannot survive. If alpha globin chain is not there, hemoglobin cannot be formed, which is a very big problem. If there is less production, obviously the hemoglobin which is going to be produced will also be less. Okay, this is one thing. Similarly, so this was the HbA1 gene and HbA2 gene. Both of them have 2 alleles. Just remember this. This is on chromosome number 16. HbA1, HbA2. Now, same thing with beta thalassemia also. You have HBB gene, 2 alleles are there. If one is affected, still it is being produced, but the amount is less. If 2 of them are affected, no production is going to happen. Got it? That means that person will be having major disease out there. Fine. So, listen to me very, very carefully. Thalassemia is a mutation or deletion reducing synthesis of either alpha or beta globin chain of hemoglobin, leading to abnormal hemoglobin and also anemia. Because if hemoglobin is not there, that means you will be having what? Anemia at the end. Low RBC count, low blood count at the end. Alpha thalassemia affects alpha globin production caused by mutation either as HbA1 gene or HbA2 gene of chromosome number 16. Beta thalassemia affects your beta globin production caused by mutation in HBB gene on chromosome number 11. This is again a blood disorder, but this is what? Quantitative disorder. That one was qualitative, and this one is what? Quantitative disorder. Here the quantity is a very big problem. Remember that difference, and that would be more than enough for you to know this. Again, carriers, 25% of chance is there. If both the parents are carrier, 25% of chance will be there for the person to be affected. That's all. So one quantitative and one qualitative is what you needed to know. That's all for this. So which one is quantitative and which one is qualitative? So this is not hemophilia, it will be thalassemia. So this is going to be qualitative because the quality of what? Hemoglobin molecule is being affected because beta globin chain is not being produced well, and this is going to be quantitative. Know that. You can get PU2 questions on the basis of this. Okay. Now let's try decoding the pedigree. Okay, so as I told in your NCERT, all the things which are explained well are recessive disorders, right? So over here you have sickle cell anemia's pedigree chart. Sickle cell anemia is what? Autosome linked recessive disorder. So whenever it's recessive, how do you spot it? Recessive disorders are not going to be found in every single generation. They will be showing skipping of generation. That means here the parents are not affected, but the children are. Here the parents are not affected, but the children are. This is what? Skipping of generation. That means that, yes, it is what? A recessive disorder. On the other side, you have a dominant disorder. If at all you see, in every single generation, you will have one parent being affected, at least one. So here one affected, here one affected, here also one affected. There is no skipping of generation in what? In dominant disorder. But in recessive disorder, there will be skipping of generation. Now I said it's autosomal. How do you understand whether it's autosomal or sex-linked? Autosomal disorders, my dear children, in that case, the males and the females are equally affected. In the sex-linked, you studied color blindness, hemophilia, males were mostly affected, right? But here in this particular case, males and females are going to be equally affected. So here, yes, male, female, here again male. There is no effect showing that males are the only one being affected. So this is what at autosomal recessive disorder, that is sickle cell anemia's thing. Now, in your NCERT, only one dominant disorder is being mentioned in the form of pedigree, and that is what? Myotonic dystrophy. It is autosomal dominant disorder. It is autosomal. Again, the male and female will be equally affected. So as you can see over here, male and female somehow are being equally affected. If at all the females are being affected more, you need not worry. Anyway, there are no conditions where the females are only being affected. It is going to be autosomal. If at all the males are being affected, you be careful because it can be a what you call sex-linked disorder also. So that is not probably going to come. It is going to be this only. So autosomal dominant trait, myotonic dystrophy is the answer for this. And over here, it is what? Autosomal disorder, and it is dominant disorder. Every single generation you can see, and males and females are equally affected, which is why autosomal. Done. Myotonic dystrophy, do not forget it. Do not forget it. I'm telling you again. It's not. Okay, yes. Now, also one thing before I go ahead with the next topic, and that is, please know that the sex chromosomes which are there, they are known as allosomes. And the normal chromosomes which are there, they are known as autosomes. Okay. Sex chromosome, how many do we have? 2 of them. Either it can be XX or it can be XY. How many chromosomes we have in total? We have 46. Out of that, 44 of them are autosomes, 45th and 46 of them are going to be sex chromosome. It can be XX, it can be XY. Cool? Yes. So if at all we talk about pairs, 22 pairs of autosomes will be there, and 23rd pair which is there, it is going to be your allosome. Got it? Yes. Now, chromosomal disorders. It can happen due to 2 reasons, aneuploidy and polyploidy. Aneuploidy means failure of segregation of chromatids during cell division results in the gain or loss of a chromosome. Whenever you have a gain of a chromosome or a loss of a chromosome, we say it is aneuploidy that is happening up over here. Why is it happening? Because of failure of segregation of chromatids. Probably the chromatids while they were separating, they didn't separate properly, which is why 2 chromatids came together over here in one particular cell. You get it? Extra chromosomes are being coming up, or chromatids are being coming up. That is because of failure of segregation of chromatids during cell division, either gain or loss. If at all there is a gain, I'll call this as trisomy. If at all there is a loss, I will call this as monosomy. What is polyploidy now? Failure of cytokinesis after telophase. So you know, first of all, the nucleus and everything divides. You have duplicated it and you have divided. Suppose the cell is not dividing, what you will be having? You will be having one extra set of whole chromosome out there. Okay, so failure of cytokinesis after telophase stage of cell division results in increase in a whole set of chromosome, and this is usually seen in the case of plants. So polyploidy in plants, PP, and here it's because of failure of cytokinesis, cell division. Nuclear division took place, but cytokinesis didn't take place, which is why you have a whole set of chromosome which is being increased. I hope that's clear. Yes, okay. Now aneuploidy, as I told you, failure of segregation of chromatids during cell division, either gain or loss. If gain, trisomy; if loss, monosomy. Now listen to me carefully. You have these three diseases. Over here, this is both of them. Both of them are going to be examples of trisomy. This is going to be an example of monosomy. Down syndrome. It is an autosomal chromosomal disease. What? Or you say it is autosomal trisomy that you see over here. Why autosomal? Because it's the autosomal chromosome which is being affected. So you know, in our body we have the 21st chromosome, so we have a pair of it, right? Suppose we get one more chromosome, extra chromosome over here. One more chromosome got added up, right? This is known as trisomy of 21st chromosome. And 21st chromosome means they are autosomes only. 23rd one is sex chromosome, right? 21st pair is going to be autosome, so this is known as autosomal trisomy. Okay. Features. This is Down syndrome, trisomy of 21st chromosome. And what are you going to see as features or symptoms? Short stature, very short height, round head will be there, furrowed tongue. The tongue will be having a little furrow like this. Broad palm crease, very broad palm crease, mental retardation and delayed development. These symptoms are super important for you to remember. Delayed development, mental retardation, short stature, round head, broad palm crease, as well as, that is the thing. This is what? Autosomal trisomy. Now there is a term like karyotyping. Karyotyping means tell the formula for the chromosomes out there. So how do you write it? You can write it like this. So instead of having 44 autosomes, generally we have 44 autosomes, right? 45th and 46th sex chromosome. So we have 45 autosomes over here. And it can be either XY. In this case, this is a Down syndrome patient who is a male. Or it can be XX. This is a Down syndrome patient who is what? A female. This is a karyotype that you can write. Okay, I'll prefer this, do not write this. Fine. Now, coming to the next trisomy condition. This is going to happen in your sex chromosomes. That means the 23rd pair of chromosome, which is why I'll call this as allosomal trisomy. Allosomal trisomy. What is going to happen over here? Usually, you have XY condition in the case of, XY condition in the case of males, right? Suppose you're getting one extra X chromosome right up there. You're getting one extra X chromosome right up there. What is going to happen? You are going to have a disease that is known as Klinefelter's syndrome. Extra chromosome is there. Here, overall masculine appearance or masculine development will be there. Remember, this is a very important point. Overall masculine development, but you will be having gynecomastia. What is gynecomastia? Breast development. You will be sterile. That means you cannot have babies. Why? Because in your body, spermatogenesis is not going to happen. Sperm formation is not going to happen, which is why you are going to be sterile. And incomplete secondary sexual development is also going to be seen. So yes, overall masculine development is there, but then you have gynecomastia, that is development of breast. You would be sterile. There is no spermatogenesis that is going to take place over here. Why? All because you have an extra X chromosome. Now, what is the karyotype for this? So here, if at all you see, the number of autosomes remains the same, 44 autosomes. Then you can write XXY. You can write XXY. This is one way of writing it. Or you can write 47, XXY. You can also write it like this, 47 as in 44, 45, 46, 47 chromosomes are there in total. So 47 and then you write XXY. So which is the extra one? You get to know it is the X chromosome out there. So either this or this. I'll prefer everyone remembers better in this way. You can write this, but in the options, if at all you're getting, please go with either of them. Cool. This is again allosomal trisomy. Now, Turner syndrome. Turner syndrome again happens with the sex chromosome. So this is an example of allosomal monosomy. What exactly happens up over here? Here, Instead of having XX chromosome, there is one chromosome which is missing, that is loss of chromosome, which is why you will have XO condition. Here, the females will be having rudimentary ovary. That means their ovary are not going to develop. I'll call this condition as rudimentary ovary, and there will be sterility. Why? Because rudimentary ovary cannot produce ova out there. Lack of secondary sexual characters, and there will be short stature. So females are going to be sterile because they have rudimentary. Reason is what? They have rudimentary ovaries. Why they have rudimentary ovaries? Because they do not have two X chromosomes out there. One X chromosome is missing. Ma'am, what is the karyotype for this? Again, 44 autosomes are there, and then XO condition. Or if at all you want to write it in the other way, how many chromosomes are there in total? 44 and 45. So 45 and then XO. You can write it like this also. Got it? This is Turner syndrome. Masculine development, gynecomastia here. Turner syndrome, rudimentary ovaries. Remember. Done. Now, Mendelian disorders, my dear children, you have these many: color blindness, hemophilia, sickle cell anemia, phenylketonuria, thalassemia. Now, in your NCERT, cystic fibrosis is also mentioned. This is autosomal recessive itself, but it has not been explained. This one, myotonic dystrophy, not muscular. Myotonic dystrophy is what? It is dominant disorder. It is autosomal dominant, the only dominant disorder which was given. Okay, chromosomal disorder, Down syndrome, Turner syndrome, and Klinefelter's. And with that, we end up this particular video, and I really hope that this helped you. Well, I took out a lot of time to explain you this, but still, if you have any doubts, please ask me. And also, I'll be coming up with a live lecture where I'll be solving the KCET PYQs separately. So do not be worried. We'll solve a lot more of the questions out there. Okay? And if at all you feel that this video was helpful for you, you understood everything, let me know in the comment section. And wishing you all the best. Do not forget to hit the subscribe button if at all you liked it. Bye-bye. Take care. Lots of love from Jasmine ma'am.